Vaccine Types and Mechanisms of Action, an Illustrative Synopsis. Pause text and illustrations for detailed study. Please refer to the previously posted Constituent Factors Observed in Viral Diseases, Parts 1 through 5, Messenger RNA Vaccination, Constituent Factors Observed, and Messenger RNA Vaccination, Immunity and Pathogenic Priming, Virus Spike Protein and Life Cycle Illustrations, Characteristics of Novel Viral Genetic Variants Possessing Viral Envelope Spike Proteins, and Spike Protein Genetics and Mutation for background data relevant to this presentation. Inactivated vaccines are produced from a killed form of the disease-causing pathogen. The resulting immunity is not as strong as that acquired from live vaccines. Booster vaccinations are generally required for the acquisition of continued immunity. Inactivated vaccine examples are administered for influenza, hepatitis A, polio, and rabies. Live attenuated vaccines are produced from a small quantity of weakened live virus, resulting in strong, durable immunity upon vaccination. Examples are chickenpox, measles, mumps, rubella, rotavirus, smallpox, and yellow fever vaccines. Messenger mRNA vaccines produce proteins triggering an immune response. mRNA vaccines do not contain live virus, causing no risk of disease to the vaccinee. Examples are some COVID-19 vaccines. Viral conjugate, polysaccharide, recombinant, and subunit vaccines contain specific pathogenic particles such as a capsid, protein, or sugar targeted to a particular viral constituent to provide a strong protective immune response to the virus. Examples consist of Haemophilus influenza type B, hepatitis B, human papillomavirus, meningococcal disease, pneumococcal disease, shingles, and whooping cough. Toxoid vaccines are produced from a pathogen's toxoid, creating immunity targeted to this toxoid portion of the disease instead of to the pathogen as a whole. Booster vaccination may be required for more durable long-term immunity. Examples are diphtheria and tetanus. Viral vector vaccines are produced from a modified version of a different virus, serving as a vector, specifically providing immunity. A DNA fragment encoding for an antigenic protein is genetically engineered into the genome of the non-replicating distinct virus, functioning as the vector. Examples are some COVID-19 vaccines. Distinct delivery vectors can be viruses such as adenovirus, influenza, measles, and vesicular stomatitis. Sterilizing immunity prevents effective host viral infection. Neutralizing antibody, NAB, provides host cell defense against an infectious or pathogenic particle. NAB binds to glycoproteins of enveloped viruses or capsid proteins of non-enveloped viruses. The neutralized particles become ineffective, infectious, pathological, and biological agents. The neutralizing antibodies function in the adaptive immune system humoral response against intracellular bacteria, microbial toxins, and viruses. Neutralizing antibody immunity is the sterilizing immunity that effectively eliminates an infection before it can occur. It does not require immune cells to neutralize antigens. Not all antibodies that engage in antigen binding are neutralizing. Non-neutralizing antibodies possess a targeting and signaling capability, providing for antigen destruction by immune cells resulting from this process. These non-neutralizing or bonding antibodies specifically bind to the antigen, not interfering with its infectivity. In sterilizing immunity, a properly vaccinated individual will neither contract full-blown viral infections nor transmit same. Mechanism of sterilizing immunity activation. The innate immune system detects SARS-CoV-2 viral RNA through interferon signaling and primes the adaptive immune system through antigen-presenting cells, APCs. The antigen-APC complex stimulates CD4 positive helper T cells, which activates B cell through interleukin 4 and interleukin 5 signaling. The B cells proliferate and differentiate into memory B cells and plasma cells that increase antibody production. The CD4 positive helper T cells also differentiate into CD4 positive memory T helper cells and stimulate CD8 positive cytotoxic T cells through interleukin 2 signaling to regulate major histocompatibility complex class 1 virally infected cell neutralization. Cytotoxic T cells also differentiate into CD8 positive memory T cytotoxic cells that together hones memory to SARS-CoV-2 that rapidly clears future SARS-CoV-2 invasion. Mechanism of action of SARS-CoV-2 messenger RNA vaccines. SARS-CoV-2 mRNA vaccine is encapsulated in carriers like lipid nanoparticles LNP to enhance cellular delivery. The vaccine is administered by intramuscular injection. The LNP mRNA vaccine enters the host cell by endocytosis, and the mRNA is released and translated by the host's protein processing machinery. The resulting protein undergoes proteasome degradation to produce peptides that get associated with MHC class I molecules. The peptides are presented by the host's antigen-presenting cells for recognition by CD8 T cells, culminating in the stimulation of cellular immune response against SARS-CoV-2. Evolution has provided for specific molecular mechanisms to effectively transport viral genomes into the host cells they infect. Viral vectors are a means by which molecular biologists deliver genetic material to cells in vitro and in vivo. Viral vector vaccines utilize SANE to deliver genes coding for a desired antigen into the host cell for another infectious agent. This enables antigen production and expression within the host cell, inducing a host cytotoxic T cell immune response. No infection with either the viral vector or the antigen source result. The nucleic acid delivered does not integrate into the patient's genome. An effective cellular immune response productive of T cells as well as B cell produced antibodies results. In the case of COVID-19 vector vaccination, the genetic code for spike proteins of the viral surface are delivered into the host cell for a productive and protective host immune response. This process mimics the immune response that occurs during natural viral infection. Basic mechanism of action of recombinant viral vector SARS-CoV-2 vaccine. DNA fragment encoding for a SARS-CoV-2 antigenic protein, such as the spike protein, is engineered into the genome of a non-replication adenovirus. Once intramuscularly administered, the DNA encoding the SARS-CoV-2 antigen is integrated into and translated by the host's protein production machinery. The SARS-CoV-2 antigen-encoded mRNA is processed by the host's ribosome to produce the spike protein, which is sensed and presented by antigen-presenting cells to stimulate a cellular immune response. Basic mechanism of action of inactivated SARS-CoV-2 vaccine. SARS-CoV-2 is cultured, harvested, and inactivated to prevent replication while maintaining its antigenicity. An adjuvant such as alum is added to the inactivated vaccine to improve its antigenicity and administered by intramuscular injection. The inactivated SARS-CoV-2 is taken by antigen-presenting cells such as dendritic cells, which undergo proteolytic degradation to liberate the SARS-CoV-2 antigen. The antigen is sensed by MHC class II and presented to cytotoxic T cells through the Th1 tumor necrosis factor TNF alpha signaling pathway to stimulate a cellular immune response. New presentations coming soon. Thanks for watching.